therapies. And what we mean by this term is that the donor and the patient are in fact the same person. The cells come from the donor, they're taken out of the body, and they're increased in cell number, perhaps manipulated to take on a certain set of attributes, and then delivered back into the same patient. So the donor and patient are the same person, and there are certain manufacturing concerns and patient outcomes that aren't relevant to autologous cell therapy. In contrast, we can compare that with allogeneic cell therapy. So allogeneic, or different gene, means that the donor and the patient are not the same person. And here the cells come from a donor that could be, for example, healthy. The cells are still manipulated outside the body, at least increased in number to reach a therapeutic dosage that is appropriate for the patient, and then delivered to the patient. And finally, the third class that we can consider is xenogenetic cell therapy. And what we mean by this is the cells come from a donor that could be the same as the patient, or it could be a different. So autologous or allogeneic examples exist in the current manufacturing practice. But the difference with xenogenetic cell therapy is that those donor cells come into contact with living cells from a non-human, from an animal. And that is part of the manufacturing process. But those donor cells are removed from the animal cells prior to administration to the patient. So even though we don't actually deliver any animal cells to the patient, because the donor cells came into contact with those animal cells, we refer to this as a xenogenetic cell therapy. Now that we've considered the different types of cell therapies, we can consider the benefits of cell therapy. First, cell therapies can be banked and cryopreserved, so you can have a set of donor cells available for manipulation and then expansion and delivery at the right time. Second, you can actually manipulate those cells prior to giving those cells back to the patient, and that provides a lot of range of therapeutic benefit and a lot of precision over the outcomes that you're seeking to induce by providing the cells as a drug. Third, you can combine these cells with other types of cells in order to lead to a positive patient outcome. So different cells can be delivered at different dosages in a mixture, or subsequently to actually have a type of combination cell therapy. And then finally, those cells can be implanted or delivered to a specific location in the body. So that potential for localized delivery is important for many specific indications. So as an example of those benefits, let's look at a particular case, human cartilage repair. So in humans, we can't make our own cartilage once it gets damaged. So when you have an injury in, for example, a knee joint, you have limited options for repair. And now cell therapy is among those repair options. In this context, what we can do is take cells from a healthy part of the patient, expand them outside the body, inside the lab to hit an appropriate therapeutic dosage, and then re-deliver those cells to the patient in a particular location, the injured site or the defect of that joint. So these newly implanted cells will then make more cells and more cartilage within the patient and then repair the defect using what the cells do best in that context. And a product that's commercially available from a company called Vericell, known as MACI, is an example of that, where we take human chondrocytes, we expand them in the lab. Through that manufacturing process, we are able to deliver a construct that addresses the defect in the cartilage. While those are very important examples, and we'll go into many more in the course of this class, let's circumscribe what we won't cover in this particular course. First, we won't cover organoids, and organoids are mixtures of cells, typically different cell types, in a matrix that has a tissue-like or an organ-like architecture and potentially some functions. So there are several shared challenges between organoid research and cell therapy manufacturing related to the source of the cells that go into an organoid, but we won't treat those explicitly in this course. Another example that is quite important, but we won't cover in this course, is using the microbiome or non-mammalian cells as cell therapy. This is an emerging and growing area of cell therapy research and development, but because this course will focus chiefly on mammalian cell culture and expansion to produce those cell products, and there are certain distinctions between how we would produce a mammalian cell product as a therapy versus a non-mammalian or prokaryotic cell therapy, we'll leave that to future discussion. So now that we've discussed what cell therapy is, the various terms that are used, the various relationships between the donor and the patient, and the indications that can be addressable, we need to dig into how we actually deliver on that promise, learn more about the cells, and learn more about how to manufacture those cells as medicine.